In this video, we're going to look at how we can modify our palmitate that we got out of beta oxidation. And we can convert it to longer chains, and we can convert it to saturated and unsaturated via elongases and desaturases. So elongases are the enzymes that make our fatty acid longer, and they're found both in the mitochondria and the ER. And they have slightly different mechanisms. The mitochondria, what's going to happen is that you have successive addition and reduction of acetyl coenzyme A units, which is basically the reverse of fatty acid oxidation. While in the ER, you have condensations of malonyl coenzyme A with acyl coenzyme A. Then you have NADPH that will do a reduction similar to our fatty acid synthesis mechanism that we just saw in the previous video. Now let's look at our desaturases, and our desaturases will create our unsaturated fatty acids, and mammals have 4 different types of these that work on fatty acids of a lot of different chain lengths. And these desaturases are delta 9, delta 6, delta 5, and delta 4. So that's telling us where we're going to put a double bond. And our enzyme catalyzes a general reaction below as shown here, where our green carbons is where we're going to add our double bond. We have variable number of carbons to the left and right. Here CH2X, X has to be at least 5, and it can contain 1 or more double bonds, while our carbons here in Y before we add the double bonds are all saturated, so there are no double bonds in these carbons. Now, when making double bonds, they're not going to be conjugated or next to each other, and we're never going to place them beyond C9. As shown in our names, we don't have any desaturase that is greater than delta 9. That means all the fatty acids that we use in our body, we cannot make, so we need to get them from our diet. Therefore, if an individual is on a completely fat-free diet, you can have poor growth, poor wound healing, dermatitis, and even eventually death. Now let's see how we make our triacylglycerides. And these are synthesized from fatty acid coenzyme A and G3P and DHAP. So our initial step is as followed. In the mitochondria, we have glycerol 3-phosphate dehydrogenase, and that's also in the ER. And in the ER in peroxisomes, we have dihydroxyacetone phosphate acyl transferase, which is going to start us off. Now, DHAP, our acyl dihydroxyacetone phosphate, is reduced to lysophosphatidic acid. So as shown here, here's our lysophosphatidic acid, and that is by a reductase that uses NADPH. So we have DHAP, acyl DHAP to lysophosphatidic acid. This lysophosphatidic acid then can be made into triacylglycerols by first we have an acylglycerol 3-phosphate acyl transferase. So now we have our glycerol and we have made phosphatidic acid. Then we can have phosphatidic acid phosphatase, which will remove our phosphate, and we'll get diacylglycerol. This diacylglycerol can then be used through diacylglycerol transferase to make our tag. So here's our glycerol backbone with our 3 fatty acids. Now you can see there's also some alternative pathways. So our phosphatidic acid can be made into our phospholipids for our membrane. Our diacylglycerol can also do that. And from eating fatty acids, we can get to monoacylglycerol, which we can use to make diacylglycerol and then be in this pathway as well. Now, the DHAP that we use to make glycerol 3-phosphate for our triacylglycerols, we get that from glucose via glycolysis that we talked about earlier in this class, or we can get it from oxaloacetate via gluconeogenesis. And so the process of making DHAP is for these pathways is called glyceroneogenesis because we're making a glycerol molecule. And during times of starvation, this is a necessary pathway because we need to make fatty acids. And actually, 30% of the fatty acids that enter our liver during a fast, we're just going to re-esterify those to tags and export them to VLDL so they can be distributed throughout the body. So free fatty acids that go in the liver get remade right into fat. So that is one of the reasons why it's hard to lose fat, is that when you break it down for energy, some of that's just going to be remade into tags and delivered right back to your cells that they came from. Also, during times of starvation, adipocytes will be breaking down their fatty acid for this glyceroneogenesis process as well. Overall, here's our big picture of lipid metabolism, where we have our tags, which can be broken down for fatty acids, and they can go to membrane lipids. Our fatty acids can go in the acetyl coenzyme A to go into the citric acid cycle. Acetyl coenzyme A is also can be used to make cholesterol. Acetyl coenzyme A can also be made to ketone bodies. Again, so acetyl coenzyme A, giant molecule because it is also fed into glycolysis, as we've seen. So everything seems to be revolving around this acetyl coenzyme A molecule when it comes to metabolism. That's it for this video. I will see you in the next one.